Hello, in this video I would describe and try to solve questions with you from IIT JAM Biological Science and Biotechnology, and the topic is immunology. So every year they ask one or two questions from immunology in both biological science or biotechnology paper. So immunology could be a very important strategy to get good marks because the questions that they ask are kind of elementary, and if you learn the basic concepts, so you can easily answer. So this is one of the very important topics, I would say. So let's move on to the questions. This question was asked in IIT JAM Biological Science 2018, and the question says, on infection by a specific virus, a host cell generates cytotoxic T cell that kill infected cell expressing self-MHC, infected cell expressing MHC of different genotype, uninfected cell expressing self-MHC, cells infected by an unrelated virus expressing self-MHC. And the answer would be infected cell expressing the self-MHC. Now I would try to explain the concept. So cytotoxic T cell provides immunity against viruses. Here is a cell infected by the virus. So the virus would take control over the host cell machinery for producing proteins, and it would produce viral proteins which would be normally degraded by the cellular mechanism known as proteasome mediated proteolysis, and some of those proteins would be loaded on class 1 MHC molecules in the ER and displayed onto the surface of the cells. That could be recognized, the viral peptide bound class 1 MHC could be recognized by a cytotoxic T cell. Cytotoxic T cell are full of secretory granules consisting of granzyme and perforin. So they would secrete granzyme and perforin, which would ultimately trigger apoptotic pathway. Now, second example, a second question that they asked in 2018 in biological science paper is, an example of transcytosis is transmission of nerve impulse from cell to cell, a pancreatic cell secreting pancreatic juice, an infant getting antibodies from mother's milk, a macrophage engulfing a bacteria, and the correct answer is an infant getting antibodies from mother's milk. So the transcytosis process is kind of similar to endocytosis and exocytosis, a combination of both. In this process, the secretory molecule traverses enters from one side and exits from other side of the epithelial cell. So here the secretory molecule is IgA type receptor, IgA type antibody. So IgA antibody, once secreted from the plasma cell, binds to the specific peptidoglycan receptor on the epithelial cell, gets endocytosed and gets exocytosed from the other side. So this is called transcytosis. As you can see, I have taken this image from Kubi's Immunology 6th edition from chapter the antigen and antibody. So they have directly taken this concept from there and asked it in format of a question. Now also, another question was asked in Biotechnology 2018, which says that antibody binds to antigen in solution through several interactions like ionic bonds, van der Waals interaction, and which one is true or which ones are true. So this is a multiple the option question, so MSQ. So multiple option could be correct. So and the correct options are all of them are correct. And also in the Kubi's Immunology 6th edition, you can see where they described all sort of interaction that is possible. Now antibodies generally exhibit non-covalent interaction. All those interactions are given there are all non-covalent and all are possible in the aqueous environment. So all these options would be correct. So as you can see, I have described that they are taking the concepts from the book directly and asking it in format of question. Now, another question was asked in 2017 biotechnology probably. So the question says, gene rearrangement in which of the following cell types lead to antigen receptor diversity in immune cell? And the options are macrophages, B cell, NK cell, and T cell. So B cell has B cell receptor, which is a membrane bound antibody. So B cell receptor generation definitely requires VDJ recombination. And also the T cell, the TCR, the T cell receptor also requires specific alpha and beta chain recombination to happen, so which is very similar to a VDJ recombination to form a T cell receptor. So basically T cell receptor and B cell receptor both need to go through rearrangement and recombination process to generate antigen receptor diversity. So this is the correct option. Now another question was asked in 2017 BL probably. Now, the cell involved in allergic reaction and containing surface receptor IgE antibodies and histamines are basophil, mast cell, monocyte, and neutrophils. So the question simply wanted to know that what are those cells which has surface receptors of IgE, that means the Fc receptors, which can bind to IgE's Fc region, and also the secrete histamine. And the correct option is basophil and mast cell. See that it's in the chapter of Kubi's Immunology's 7th edition in the allergy and hypersensitivity reaction chapter. So where they have given that what cell secretes what type of cytokine and everything. So mast cell and basophil both has Fc receptor. And I have a separate video on how this mast cell basophil functions in case of an allergic response. I would recommend you to watch that. Now, a question in IIT JAM Biotechnology 2016 where they asked a pure IgG antibody was run on a SDS-PAGE under reducing condition. How many bands would you see after staining with a Kumasi blue? And the answer is 2. The reason being, so you can see here is an antibody and there are disulfide linkages in blue, and in a reducing condition when you are giving SDS-PAGE, these disulfide bonds would be gone. So basically you would get kind of 4 chains, 2 heavy chains, 2 light chains. Now since the heavy chains, 2 of the heavy chains are of same molecular weight, so they would appear as 1 band, and 2 light chains, since they are of kind of similar molecular weight, they would appear as another band. So you would ultimately get 2 bands in a Kumasi blue staining. Another question was asked in 2016 BL or BT, maybe 2016 BT, where it asked that identify the autoimmune disease among the following. And the options are type 2 diabetes, type 1 diabetes, and gestational diabetes, and pernicious anemia. And the correct option is type 1 diabetes mellitus. Again, this question was taken from another table in chapter, the allergy and hypersensitivity chapter. So you can see there is a list of diseases given where type 1 diabetes mellitus is actually autoimmune diseases, where the Th cell actually activates plasma cell to produce antibodies against the pancreatic beta cells and create severe inflammatory responses and all these things. So this is again taken from Kubi's Immunology. So they are over and over every year kind of taking concepts from Kubi's Immunology and asking questions directly from the book. Another question was asked where it says antigen binding site of an antibody molecule consists of an N-terminal domain of heavy chain and C-terminal domain of light chain, N-terminal domain of heavy chain and N-terminal domain of light chain. So anything like that. So the correct option would be this, because you can see if here is the antibody, and here would be the N-terminal region of the heavy chain, N-terminal region of the light chain. So the antibody binding groove is present here, the hypervariable loops, if you remember. If you want the details about this, you can watch my videos about antibody structure and function. So I hope you enjoyed this video and this video was informative. So I would give all the relevant concepts along with these videos in the descriptions. And if you want to learn more about immunology, I have a immunology playlist which you can refer. So I hope you enjoyed. Please like, share, and subscribe. Thank you.